Okay, well, welcome. Uh, Thanks for being patient last week. I appreciate very much Rita's helping us out last week. And this, I believe, is our ninth week. And today I would like to spend most of the class analyzing the toggle circuit, which is an important motif in especially developmental biology. As always, I need to remind you that this is recorded, edited, a version is made available. So today we're going to just very quickly go over some of the things that were discussed last week and briefly mention limit cycles, which I don't think Rita got to. And then we'll talk about functional declarations and maybe do an exercise or two, quick exercise or two on those. We've talked about them a little bit, but I don't think we did any exercises on them. And then we will dig into the toggle circuit. And I don't know how much of it we'll do. There are a lot of exercises one can do on the toggle. And I realized when I was putting this lecture together that I had used two different mathematical forms of the toggle. So some of the exercises use one mathematical form, some use the other. They both behave very similarly, but the parameter values are a little bit different. So I appreciate your patience. You could help me maybe harmonize the deck afterwards. And then I'd like to talk a little bit about Gillespie stochastic solvers for these kinds of networks, if we have time. And the toggle is a place that specifically you get qualitatively different results when you do the stochastic simulation from what you do when you do the deterministic simulation. Usually the stochastic simulations are rather similar to the deterministic simulations with noise. But in the case of the toggle, you actually get a qualitatively different result. And that actually has quite a bit of importance in the context of developmental biology. It means that gene regulatory circuits by themselves can't explain things like differentiation. So last time, Rita kindly went over the basics of dynamical systems theory in 2 and more dimensions. We had talked about the basic concept of a fixed point, which doesn't change when you have a difference dimension. It's the same in any dimension. But you have to generalize the concept of a perturbation and say that for all perturbations that are sufficiently small, the amplitude of the perturbation goes down if you want to have a stable fixed point. And for at least some perturbations, the amplitude of the perturbation goes up if you want to have an unstable fixed point. And that isn't a particularly challenging concept per se, but it does require a little bit of linear algebra to make that mathematically precise. And so here I've started out with a fixed point and star. I have a perturbation epsilon. That takes me to a new initial position at 0. And now I repeatedly apply my time evolution. And that epsilon will evolve in time as time goes forward. And I want the amplitude of epsilon of t. This really should have a subscript, probably epsilon 0 here and epsilon of t here, if I were going to be more clear. Epsilon should go to 0. The length of that should go to 0 as time goes on, if I want to have a stable fixed point. Now, here's where the linear algebra comes in. If I have a fixed point n star, and dn by dt is some generalized vector function of n. Then if I linearize around that fixed point, then, and I take the Jacobean matrix, the derivative of the function in the n dimensions, then if the eigenvalues of that Jacobean are lambda I, the fixed point is linearly stable. And I don't know why this says the trace of the Jacobean matrix here. That's a typo. It's probably a cut and paste typo. The fixed point is linearly stable only if and only if the real part, some of them potentially are imaginary, so, or complex, and so you could have complex eigenvalues. The real part of all of the eigenvalues is negative. If any real part of any eigenvalue is positive, then you have an unstable fixed point. If the real part of all but all of the eigenvalues is negative or zero, then you have a marginally stable fixed point. So if you have any zero valued, zero real component of the eigenvalues, you have a marginally stable fixed point if the others are negative. And that's true in any number of dimensions. One of the things that tells you is that as you go to higher dimensions, it becomes easier to have unstable fixed points and harder to have stable fixed points, because you need to have, if you're in n dimensions, in principle, if you don't have degeneracies, you have n eigenvalues, and all of them have to be negative. And so the possibility of one or more positive eigenvalues goes up, and also means that while the stable fixed points don't really change when you go to higher dimensions, you get additional types of unstable fixed points. You could be stable in 2 dimensions. To be stable, you always have to have all of your eigenvalues negative. So you have inward direction along the eigenvectors, and your unstable possibilities are you have inward in one eigenvector direction, outward in another. That's a saddle, or outward in both. It's called a star. In 3 dimensions, you could be inward in 2 directions and outward in 1, inward in 1 direction, outward in 2, or inward in no directions, outward in 3. So you have additional unstable fixed point types. In general, we tend to focus more on the stable fixed points than the unstable fixed points, but there are additional types of unstable fixed point in higher dimensions. The theorem that the real part of the eigenvalues has to be negative tells you that if the trace of the Jacobian matrix is positive, then the fixed point is unstable. Why is that? Because the sum of the eigenvalues is equal to the trace. And so if the sum is positive, then at least one value has to be positive. Doesn't tell you if the sum is negative, you could still be unstable because you could have positive one outweighed by more negative ones. But if the trace is positive, you're definitely unstable. And why do you try to define rules like that? You try to define rules like that for 2 reasons, one of which is that it winds up you can classify the fixed points based entirely on the trace and the determinant of the Jacobian matrix. So you don't have to look at all the eigenvalues directly. You don't have to evaluate the eigenvalues, which sometimes is numerically or analytically a pain. And it allows you to actually make that nice chart of the pattern of stability. So again, in 2 dimensions, you have a relatively simple classification. Again, you can have two positive eigenvalues. You can have one positive and one negative, which is saddle, both negative, which is stable fixed point. In 2D, you can evaluate the eigenvalues if it's non-degenerate. Analytically, it's just the quadratic formula. Lambda plus minus is trace of m, square root of twice squared, minus 4 times the determinant divided by 2. Notice that trace squared could be less than 4 times the determinant, in which case you'd have a complex result rather than a real result. If the determinant is positive of the Jacobean matrix at the fixed point, then you can do a general classification in 2D, and you get a picture that looks like this. And there are different ways of organizing the choices you have to make, the hierarchy that you have to make to get your different regions. But the first question is, if the determinant is negative, then you have an unstable saddle. You have one positive, one negative eigenvalue. And so here on the y-axis, I really should have a computer-drawn version of this rather than a hand-drawn version of this. I guess that hand-drawn version is probably 30 years old. I just propagated it. Here's the trace in the x-axis, the determinant of the Jacobean matrix in the y-axis. If you're negative, it's all unstable, it's all saddle. If the trace is, if the determinant is greater than 0, I'm up on the top, I have 1, 2, 3, 4 regions plus the boundary lines between them. And this boundary is determined by trace of squared equals 4 determinant of A. That is the argument of the square root is either positive or negative. And so if trace squared is bigger than 4 times the determinant, this argument is positive. All my eigenvalues are real. If this is negative, then I have complex eigenvalues. And so I now ask the question, if the determinant is positive, I'm on the top here, I now ask, is the trace negative? If the trace is negative, that's the only quadrant in which I have stability. And so here I am in the stable region. If the trace squared is bigger than 4 times the determinant, trace squared is bigger than 4 times the determinant, in that case, I have two negative real eigenvalues. I have what's called a node, stable node down here. If the trace is equal to 4 times the determinant, then the argument is 0. I have two degenerate equal eigenvalues, and that's called a stable star or improper node. Basically, it's one-dimensional. If the trace squared is less than 4 times the determinant, I have a complex conjugate pair of eigenvalues, and that gives me what's called a stable spiral. We'll come back to that in a minute. If the trace is 0, I'm on the center line, I have what's called a center node. That happens to be a neutrally stable state. If the trace is positive, then I know I'm unstable. I'm now in the top right region. And again, if the trace squared is bigger than 4 times the determinant, my eigenvalues are both real, in which case I have a node. If the trace squared is equal to 4 times the determinant, then I have degenerate eigenvalues, which are both positive, which is a star improper node unstable. If trace squared is less than 4 times the determinant, I have an unstable spiral. And so if I want to quantify a 2D fixed point, I don't actually have to solve the eigenvalues directly. I solve the determinant of J star. If it's negative, I have unstable saddle. If it's positive, I look at the trace. If the trace is negative, I'm stable. If it's 0, I'm center I'm positive it's unstable. And then I compare trace squared to 4 times determinant. If trace squared is bigger than 4 times determinant, I have a node. It's equal, I have a star, or improper node degeneracy. If it's negative, if it's less, I have a spar. So I can quantify everything without actually doing all the finding the eigenvectors and eigenvalues. Now, this idea of a spiral introduces us to what are called limit cycles. And we start out by asking the question, what are, if we have a fixed point, what happens? But there's an additional complication in 2 and higher dimensions, which is we showed that in 1 dimension, you can't have oscillations. But in 2 or higher dimensions, you can have stable or unstable oscillations. And those spirals tend to lead to are oscillatory in nature. An unstable spiral represents a growing oscillation. Stable spiral represents a decaying oscillation. If you have an unstable spiral, it could lead to something else. It could go to a different fixed point. But it's also possible that it leads to what's called a stable limit cycle. A limit cycle is something where a situation in which you have temporally periodic behavior of your system. And the simplest way to deal with limit cycles is to go into polar coordinates. And they all have the same, we did the simple forms for the various bifurcations in 1D before. A limit cycle has a simple generalized form. In this case, the radius of the limit cycle, if I think of a circle, the radius of the limit cycle is exponentially coming back to whatever its target radius is. So the radius goes like dr dt is alpha r minus r, capital R. That's going to be 0. The value of that is 0 when little r equals big R. That's called an invariant cycle or orbit. And then I have an angular component, d theta dt equals omega, which is the frequency of the oscillation. In this case, if alpha is negative, the limit cycle is stable. If I move away from it in R, I come back. If alpha is positive, the limit cycle is unstable. I move away. Notice that I'm not asking for stability in the phase. If I move away in theta, I don't come back in theta. I'm not asking for that. There are situations like phase locking where you can have the phase also be entrained, but we're not doing that here. Okay. If I want to convert this back into Cartesian coordinates, it doesn't have a particularly pretty form. Du by dt is alpha times u, and that alpha, remember, is going to determine the stability of the limit cycle, minus v minus u times u squared plus v squared. So u squared plus v squared is the radius squared of the coordinate. So that's maybe helps you think about what that means. And dv by dt is the same thing with u and v exchanged. And you could put that into your collab notebook and see that it behaves like that. So again, it's simpler in polar coordinates, but we do most things in Cartesian coordinates. And if we then plot this, here I've plotted the vector field of the flow at each point, and here I've actually plotted trajectories. And I see if I look in the middle, start in the middle and move out a little bit, I spiral out. If I start outside of the limit cycle, I spiral in. And in this case, the central fixed point is unstable. The limit cycle itself is stable. In the other one, which looks very similar, you'll notice that the central point is a stable fixed point. The limit cycle here is in fact unstable, and I spiral away from the limit cycle. What happens at infinity, I don't know. I'd have to have something else to see what happens. But in this case, there are these two things. You'll notice that if I actually look at these two pictures, if I look at the vectors, I can see in one case they point inward, and in the other case they point outward. But if I look at the actual orbits, if I don't know the direction in which time is moving, I can't tell the difference. They're symmetrical.
Oh, I have a question. In the case where alpha is a -1,
Yes.
if the point starts on the orbit, it will either move out or move in the circle, right? And
Yes.
if it's moving inside, it seems it will eventually end in another stable point, like the center of the circle.
That's right. There's a stable there. That's right. There's a fixed point here when r equals 0. So it stops.
Is it is
Uh,
it reflected by the formula, by the equation?
well, you have it. If we look, let's see. It should be. Let's see.
Oh, okay.
If I make u and v both 0, and it's always a little bit hard to do this analysis in the polar coordinates. So when we're going to do if we're going to do the fixed point analysis, it's easier for the fixed point analysis. It's easier to use the Cartesian coordinates.
Okay, so the analysis of circles and the analysis of fixed points, they are separate.
You can Yes, you can do both, either in polar coordinates or in Cartesian coordinates. But the cycle, it's a lot easier to analyze the cycle in polar coordinates, a lot easier to analyze the fixed points in Cartesian coordinates. That's why I wrote the equations both ways.
Okay, okay,
But if I look here and I set u and v to 0, then the right-hand side of both of these is 0. So 0 comma 0 really is a fixed point.
okay.
And let's look at stability. If I take the derivative of this of du dt with respect to, well, then I have to do the full analysis. But I'm going to find effectively that the eigenvalue, the real part of the eigenvalue is alpha.
Mhm.
And so, actually, minus alpha, because I put a minus sign here. I changed the sign here relative to the other one.
Okay.
So I have alpha minus alpha. So alpha being positive is stable, alpha being negative is unstable for this picture.
Hmm.
Good point. Okay. And so that's all that we have really in the way of fixed points. There are, as I mentioned, there are higher dimensional uh, orbits and structures which you can run into. Uh, we're not going to handle that. I wanted to come to a little bit of antimony, some very simple antimony. The first one is going to be the concept of a functional relation. And I'd like you to try this out in Tellurium using Colab. And we'll start out by just writing a very, very simple chemical reaction, the shortest thing that I can do. So you don't have to type, you don't even have to name the reactions. And you certainly don't have to have the comments. A goes to B at a rate K1 times B. B goes to nothing at a rate K2 times B. Start with A equals 10, B equals 0. Set K1 equal to 0.2, K2 equal to 1, for example. And the reason for this is antimony specific. We're used to multiple kinds of equality in, say, Python or C++, where the equal sign that does does comparison is a different form from the equal sign that does assignment. And in antimony, we're going to have two different kinds of assignment. We see here, for example, under species initialization, A equals 10, B equals 10, K1 equals 0.2, K2 equals 1. And if we load this antimony model and change and have different values of A and K1, that's fine. However, if we go in and we overload these parameter values, we'll find that they may or may not behave exactly the way we expect. These equal signs are evaluated once at the beginning when we load the model, and they're not looked at again. And so if I said A equals 10, B equals 2 times A, and I change the value of A in Python, A will change, but B will not change. And so functional relationships and time-dependent relationships are a way of dealing with that. And we've already talked about it, I think, when we talked about chain variable stoichiometry, because we had to use it for that. But I just want to come back to it now for a minute.
Oh, there's something wrong with my programming environment. Just wait a minute.
Okay, let me check. I'm just running it. I'm just running it myself. It took a while for the pip install to run, but it ran for me.
Some library might be broken. I'm still trying to fix that.
You're running it. Oh, but you don't use Colab. You're running it local, right?
Yes, I'm running it locally.
Okay. Unfortunately, that I can't help you. That was the big reason we went to Colab, was because
Okay.
uh, uh, so that when there was a Python update or something like that, it didn't break people's code. Pedro, have you seen a problem with antimony and tellurium lately?
What do you mean?
Well, Chen is saying that he's having an issue running Tellurium on his local install at the moment.
I never tried that. I only use CC3D, Tellurium, and Colab. So it could be a version problem,
Oh, I
but I
fixed
don't.
it. Oh, I fixed that.
Got it? Okay. So this is about the simplest thing we could have. So um, I just want to have some reaction that we can play with. And if we already did this, let me know, because it's possible we already did this and I forgot about it. So does this work? Do you have this one working now?
It's running, but I'm not getting the same figure.
Oh, do you want to show me what you're getting?
I think I think some parameters might be wrong.
That's okay. The parameters don't matter. But if you want to screen share, I can take a look.
Oh, I can share the screen. It's yeah, here it is.
Okay, let's see what you get.
Okay, so results will don't need that.
Yeah, you don't need anything very fancy here, just
Yeah.
plot.
And let's plot the A and B.
So you just do t dot plot, that'll do it.
Oh yeah, sure. Right. It's a result dot plot, right?
Well, t dot plot will plot whatever the last thing that was calculated is.
Okay, sure.
Not very pretty, but it's the fastest way to do it.
Wait, what? Plot requires
Oh, because you don't specify what your output is. In line 17, just say r dot simulate t dot, yep. No, but you don't, no, get rid of the time AB. Yeah, get rid of that.
that's it.
Yeah, that's it.
What?
What? By position arguments. R dot simulate. Hold on a sec. Can you scroll up, please?
Scroll up.
Up, just to see the beginning. The single t dot r load A, fine. A goes to B, B goes to A. Okay, that looks okay. Simulate. Why don't you just print set result equals r1 dot simulate, and then let's just look at it. I don't understand why. Why is it? What's it giving that error? Let me look.
I can plot it using
Oh, sorry. I'm sorry. I said t dot plot, and that's wrong. It'll be t dot plot array. It's just r1
right.
dot. It is r1 dot plot as you were typing it. Yeah.
This one.
Or just type r1 dot plot.
Oh,
Yeah, there you go. Just plot. There you go. That's it.
oh yeah, right.
Okay.
That's it.
Sorry about that. The syntax is, I don't know why. Well, I do know why, but if you use plot array, because then you specify the argument of plot array,
Mhm.
it's te dot plot array. But if you're using the plot, it's just r dot plot. So, okay, fine. So now we're going to put some modulation on one of the parameters. K1 of t is 0.12 times plus sine of t, for example. And to do that, we use the colon equals syntax. And this is telling us that we're going to, the colon equals says that this equality is continually reevaluated.
It seems that the slides are not displayed.
Oh, sorry about that. Yes. No, thank you for telling me about it. Is it back?
Yes, it's visible.
That's right. So here we want K1 now to be a function, the rate of reaction to be a function of time, for example. I will just pick some arbitrary thing like 0.2 plus sine of time. And we're going to use the syntax colon equals. Regular equal sign is evaluated only when you do the load A. Colon equal is updated continuously in time. And so if I say K1 colon equals 0.2 plus sine of time, here, replace the K1 here by that, try that. And you should get something different when you run it. Yeah. There you go. And now instead of typing colon equals, just type equals. What do you get? If you type equals, what'll happen is it'll evaluate 0.2 plus sine of time at time equals 0. Sine of time equals 0. Sine of 0 is 0, so it'll just give you 0.2. You should get what you had before. Does that work?
Yeah, it's the same figure.
And so the colon equals is fine. The one thing about a colon equals is that you can't overwrite it from Python. At least you used not to be able to. So I cannot set the value of K1 anymore from Python. It's privileged. So here's what you'll get now. You could also make K1 a function of, for example, a variable. You could make K1 colon equals 0.2 plus sine of A. Don't have that as an exercise, but try it and see what happens. Did it work?
I'm not sure what to edit here.
So just replace the argument of sine. Here I have sine of time. Make it sine of A, capital A, the species. So we often make these things time dependent, but they could be dependent on a variable instead of on time. That's allowed. In this case, A is decreasing in time, so it's going to give you something rather different. Yeah, okay. Not so exciting, but it runs. Okay. Now let's say K1 is increasing in time. So we could say K1 of t is equal to 0.2 plus 0.1 times t. We'd write that K1 colon equals 0.2 plus 0.1 times time. So try that one. Yeah. Okay. Yeah. There we go. So the reason that this becomes more significant is the following. Suppose that I want to have a lot of variables that are scaled. So I want to have, say, a length scale, a time scale, and I want a bunch of things to depend on that time scale. I might do something like this here. K2 equals 3 times K1. That's fine. But if I write K2 equals 3 times K1 and I change the value of K1, this will be evaluated. If I change K1 in my code, K2 will change. However, if I go into Python and change the value of K1, K2 will not change because this equal sign is only evaluated once when the model is loaded. And so if I want K2 to change when I change K1 from Python, I would have to write K2 colon equals 3 times K1. Similarly here, if I look in the code that I've got on the screen, I have K2 equals 1. Actually, this is wrong. I don't want that. That K2, that first K2 shouldn't be there. That's a typo. I've been using that slide for years and I never noticed that typo. K1 colon equals 0.2 plus 1 times 0.1 times time. K2 equals 3 times K1. If I write that, K2 will just be equal to 0.6. K2 will not increase in time. If I replace it with a colon equals, then K2 will increase in time. Does that make sense? Again, if you use a regular equals, everything works, but only when the thing is loaded. If you want it to be updated in time, either from changes of value within the simulation or from the Python layer when you're doing something in the simulation, you have to use the colon equals. The other thing that I was going to mention, I think I may have mentioned already, is that if you want to write ordinary differential equations directly, rather than using that nothing goes to arrow notation, I just use prime sign. So if I write K2 prime equals 3 times K1, that's equivalent to writing dk2 dt equals 3K1. And so that will also update continually in time. If I do that, and I write K2 prime equals something, then it's required that I specify the initial value of K2. So that would be like a variable. Basically, I'm writing an ODE, like a rate. I'm writing another rate. I don't think we have to do that, but here I have K2 prime equals 3 times K1. And here I do need that K2 and it equals 1, or whatever the initial value of K2 is. And so these are additional ways of specifying time dependencies in these simulations that you may find useful. All right, I'm not going to do that exercise. I don't think it's that exciting. If I run the simulation as a whole without specifying which things I want to output and use t.plot, r.plot, I'll get extra lines. If I specify K1 colon equals, it will treat K1 as a variable and plot it. If I set K2 prime equals something, it'll treat it as a variable and plot it. So in that case, I'll have to use the column selection to get what I want. All right. So I have to use the column selection, which you already were doing in your code. All right. So I wanted to come just to review those, because for the toggle, it may be convenient to write things that way. And so I want to come back to this circuit. A couple of weeks ago, we analyzed an oscillatory circuit that looked a little bit similar to this. We had an auto activation loop, and then we had a B species which interacted with A. In this case, we have A promotes itself, B promotes itself. So we have two, and remember, auto activation is bistable. And then we have A inhibits B and B inhibits A. And this circuit, to begin with, is completely symmetrical. A and B don't have any distinctive features. They could swap A and B to wouldn't change it. Now this, when it's representing a gene regulatory network, is a fairly common motif. A classic example of this is in early embryos. There are a pair of transcription factors, one of which is called SOX2 and one of which is called OCT4, that mutually inhibit each other and determine whether a given cell is going to be part of the embryo proper or the extraembryonic tissue. And so all the cells will either have high SOX2 or high OCT4. And that's going to determine the cell fate. And so when we start out looking at this, and I have a variety of different ways of walking through this exercise, and we'll see how we go with this. So I say I took two versions of it because there was some additional material that I wanted to cover, but we may find some inconsistencies in it. So I apologize in advance for that. So it's called, why is it called? First question is, why is it called a toggle? I think I've already explained that to some extent, but we can look at that. And then what are the reactions in the model? And the first thing we can think about is, and this is thinking about, we didn't really do that this year, but there's a whole approach to analyzing these networks. This was called Boolean networks, where you say that the values of A and B are not continuous, but just 0 or 1. And so then you treat these as logic circuits, and you time step the situation. So you say, for example, here, you would have either an and or an or between the signal from A and the signal from B, either an and or an or from signal from B and B. You set initial values of A and B. You evaluate what the next state would be if those were the initial states, and you do time stepping on them. That's called the Boolean network approach. It has the advantage that it's very, very fast computationally because you're only doing single logical operators. You're not solving differential equations. And it actually, for gene regulatory networks, it often gives answers that aren't too far from reality. And so let's think about the input signals coming into A. A has two signals. It has an activator coming in from itself, an inhibitor coming in from B. So if A is high and B is low, what are the input signals to A? If A is high, the autoactivation is high. If B is low, the inhibitory signal is activating. And so I have two activating signals coming in. So that's a consistent signal. Similarly for B, if B is low, the activation of B on itself then is low. A is high, so the inhibition of that is low. And so I have two low signals coming into B. So if I start out with A high and B low, A stays high and B stays low. What happens if A is high and B is high? Is the input consistent or inconsistent?
It's inconsistent.
Right. Because the autoactivation in both cases is consistent. A is high and it's being activated, so it stays high. But the cross signal, the cross inhibition is inhibitory. So I have what's called an incoherent network. A is trying to activate itself, but B is trying to inhibit it at the same time. So if A is high and A is low and B is low, A activates A is 0, B inhibits A is 1. So that's an inconsistent signal. If A is high and B is low, A activates A as 1, B inhibits A as 1, so that's a consistent signal. So, and if I did a Boolean network, I could get oscillations that way for the inconsistent ones. If I start with a 1, 1, then I can get an oscillation. Okay. So the next thing that I want to do is to write differential equations for this circuit. And this was actually a homework problem. And so if you did that homework problem, then you can just look back at this. And the other thing that I'm going to say again is that there are two Herbert, in his homework problem in the textbook, used a different functional form for these equations from the one that I did in the lecture. And they both give very similar results, but they're not quite identical. And I wrote them the way I did because it's a little bit easier to do the analysis, but fundamentally they behave the same. And so what I want to do is first go over how to go from the diagram to the differential equations. And then we'll plot a time series for this. for just one set of parameters and initial values. And then we can plot A versus B. That's actually a rather interesting one to do in this one. And we'll repeat for different values of A and B and overlay it to make a phase portrait. So do you remember how to go from a diagram like this and try to write differential equations for it? Do you want to try doing that on your own, or do you have it ready? Is that a useful exercise? You tell me.
Yeah, I have it already.
Okay. So if you have it already, let me just review it, because I think it's always worth thinking again about how you get there. So this arrow diagram here really represents the gene regulatory network. And here I have the gene A, and here I have gene B, and I have two promoter regions, one of which is a positive promoter, one of which is a negative promoter. So if I want to write this in SPGN notation, the notation you use to describe gene regulatory networks, I guess it looks like this. And I have to remember that A always decays, the protein always decays. In this case, the messenger RNA decays. And in this case, I'm also neglecting the translation of messenger RNA into protein. In some cases, the messenger RNA itself is the thing that's going to do the signaling. But that's depends on the situation. And so that very abstract high-level diagram then turns into this more detailed description of what's happening in the molecular circuitry of the cell. And then the base concept here is that I'm just going to look at the auto activation. A activates A, B activates B. If A activates A, I'm going to say that its activation is a Hill function of A, an increasing Hill function of A. So here, A to the H over K1 to the H plus A to the H. There's a basal rate of transcription, B, and there's a decay term minus K2 times A. And I write exactly the same thing for B. And so that's writing A activates A, B activates B, and ignoring these cross inhibitors. And here I've made things symmetrical. I've assumed that all my rates and levels are the same for A and B, which might or might not be true. Now for cross inhibition, I'm going to assume that it has an AND form, product form, and I'm going to assume that the inhibition works like a decreasing Hill function. So I have the inhibition of A by B goes like, again, K1 to the H over K1 to the H plus B to the H, and vice versa. And I'm also going to assume that there's some strength of inhibition. And I'm going to say that the inhibition is maximal if this value parameter I is 1. And the two circuits are decoupled if I is 0. And so then my modulator, I want my modulator function to be between 0 and 1. And so my modulator function is going to be 1 minus I plus I times my Hill function, because my Hill function is between 0 and 1. And so this is not perhaps it looks a little bit complicated when you write it this way. But again, think about the limits of this. If I is 0, then I get 1. And so if I is 0, I have no inhibition. If I is 1, 1 minus 1 is 0. I here is 1. I just get K1 to the H over K1 to the H plus B to the H, which is my inhibitory Hill function. And I've asserted an AND form. So all I'm going to do is multiply the activating term here by the inhibitory term. So I wind up with something that looks like this. And I realize again that this looks like a lot of mathematics, but it's actually not bad to analyze it. And I want to, now we built it. And when you build it in steps in a systematic way. But a lot of times when you're reading a paper, you'll see a set of differential equations, some long set of differential equations. And it's useful to build some intuition about what those differential equations mean. So when you see some long string of differential equations for chemical species in a cell, it's not something that in general can't be understood. So occasionally they'll have some strange functional form, which takes a lot of work. But for example, here, suppose that I hadn't walked you through, I didn't have the diagram. You didn't have the diagram of the circuit. And I just wrote the final form of these differential equations, which is not, it was relatively elaborate. And so if I saw these without building it up and it wasn't annotated properly, it could be confusing. So let's just think about it now for a minute. And I walk through step by step what the terms mean. So the first thing that I notice is that there are these terms B and B here, that aren't necessarily multiplied either by A or B. So this is a constant. It really is KB times 1 minus I is a constant. And that means that as long as I is not 1 and B is not 0, that there's always some production of A and B, no matter what's happening. And so that represents Basel production. And that would be true always in these kinds of gene regulatory networks. They wouldn't necessarily, you wouldn't expect Basel production as signaling network. So that represents this arrow, part of it, effectively this arrow here. The next thing I have is that activating Hill function, a to the h over k to the h plus a to the h. Which represents the self-activation of A and B. And that represents this activation here, binding to that positive transcription of that positive regulation site. K represents the maximum rate of production of the species. And I'm missing a purple label here on the arrow. I have first order decay kinetics, minus K2 times A minus K2 times B. And that's almost always going to be the case in almost any equation you run into. Occasionally, you'll see Michaelis decay kinetics or activated decay kinetics, but in general, first order decay kinetics. So that's here. I have that inhibitory function, K over K plus B. That's the inhibitory line here. And then the last thing that I have is that I have to know how the activator and the inhibitor combine. And here I'm multiplying the activation, the inhibition. That has the form of an and. That means the inhibition wins out over the activation. And the last thing is that I have these yellow arrows, which regulate how strong the cross inhibition is. And that's the 1 minus I plus I here. And so each term in this ODE corresponds to a specific biological function, and you can tie it specifically to this kind of diagram. And it's worth going through a bunch of papers and just systematically walking through term by term the ODEs and doing this kind of analysis and developing a little bit of an ability to translate. I prefer the arrow diagrams because those can, if they're annotated properly, unambiguously give you the ODEs. The ODEs, it winds up, can be ambiguous. There could be multiple circuits that give the same ODEs. But it's good to learn how to translate both directions. All right. So if we look in Herbert's textbook in the homework, you'll see that I combined the A and the B, activator and inhibitor, in a slightly different way in that. Here, the rate of production of A is K1 times A to the H over the Michaelis or Hill constant to the H plus A to the H plus B to the H. Notice that this is also activating in A and inhibiting in B. And it also has the property that its maximum value is 1, K1, and its minimum, I've got to be careful because he doesn't have the K. So its maximum value is actually K1 divided by K A to the H. So it's not normalized in the same way that this one is. But they are slightly different. And if you look at what happens when A and B are both non-zero, their shape of the curves is a little bit different. And so Herbert gives a particular set of parameters, 2.2.68 for the Hill exponent, Ka5, Kb5. These scales A and B to 10. Here's a question, and I have a solution for one solution for it. And we won't play around with it too much, but if you wanted to have variable strength inhibition in Herbert's form of these equations, how would you do it? What would the simplest way of introducing it be? I'm going to modify these equations here just a little so that instead of having maximal inhibition between A and B, I can control the strength of the inhibition. What would I do? There are probably a couple of ways you could do it, but there's an obvious simple way to do it.
Maybe add another parameter.
Right, we'll add a parameter. Suppose there's a parameter alpha that is the strength of the inhibition. And so if alpha is equal to 1, the inhibition is maximal. And if alpha is 0, the inhibition is non-existent.
Mm.
What do I have to do, for example, this rate law? Where do I put alpha in that rate law?
It's the strength of in front of B.
Yeah. So if I multiply B by alpha, then if alpha is 1, I get back my full inhibition. If alpha were 0, I would have just K1 A to the H over K1 K A to the H plus A to the H, which would just be the auto inhibition. Yeah. So if I put alpha in front of B, alpha in front of A here in the second one, then that has the same effective form as this one down here. This is another exercise where perhaps we don't have to do it, but I want to talk about it. This was one that originally I wrote for this set of equations rather than the other one. Something that is useful to do when you're starting out, when you don't know how the system is going to work, is to run it for an initial set of initial values of A and B. For example, 0.1, 0.5110. And then see what the typical output values of A and B are. Don't run it for very long. Just run it for a short amount of time. Now, in this case, if I look at A and B, I know that these hill functions are going to effectively confine the values of A and B to being between 0, they're always positive, and the hill function switches when A is equal to K, and it saturates when A is about twice K. And so if I've set Ka and Kb equal to 5, I expect A and B to range between 0 and 10. I'm allowed to have A and B be as large as I want, but the typical values will be between 0 and 10. I have to be a little bit careful because when these decay coefficients are large, it'll pull down that value. And so something that's useful if you come up with a set, here I know what I've done, but if I were faced with a set of equations from some paper, and I didn't know a priori how they behave, it's worth initially running the simulation for a couple of different values of A and B to get a sense of what the possible ranges of A and B are. And now we come back to the next question, which is, suppose that I run this for a bunch of different conditions. If A is larger than B, what's going to happen? Initial A is bigger than B, what's going to happen? Based on the analysis I already did.
A will get larger and B will disappear in the end.
That's right. Now, if I start with A equal to B, I get stuck. I'm on an unstable fixed point. And then there are these questions, are there initial values when you wind up with either small a and small b, or large A and large B? Yeah. That's generally not going to be the case. On the other hand, if I start out with A equal B, that can happen. So why don't we just run this simulation? I think you did this as a homework problem, so you have the simulation, probably have the simulation from a couple of weeks ago.
Yeah, I did that.
Okay. So if you can you get it running again?
Yeah, I still have the code.
Okay, so I'll give you a second to just pull it up and run it. Let me know when you got it.
Okay, I got it.
Okay. So now what we should see is, depending on our initial, here's just the time series for X, for example. If I start with X, well, here X, I called it X, but A. If I start with one high and one low, if A is bigger than B, A stays high, and B goes and if A is less than B, A goes low. And so this was an exploration of the different situations. It's actually pretty straightforward here, in the sense that The only thing that matters is whether A is bigger than B or not. And maybe since you've got it, we don't really need to do it. But what I asked you to do in the homework was, for example, fix the initial value of B, say to be 0.7, and then sweep the initial values of A and see what happens. And of course, you should get something that looks like this. I think you probably remember this. And here, I apologize, I've switched back to my equations rather than Herbert's. Because they're scaled. These are scaled between 0 and 1, which I prefer. And so if my initial value of B was 0.7 and my initial value of A was bigger than 0.7, A goes to a high value. If my initial value of A was less than 0.7, A will go to a low value here. And there is some, and if my initial value of A was exactly 0.7, A and B stay equal to each other, but they actually don't stay constant. They both will move to give a value of a half to balance that. And similarly for B. And as I see, if I have A high, I get B low. If I have A low, I get B high. Notice that the value of A and B go to 1. The low values are not 0. Why are they not 0? They're not 0 because I fixed the value of I have a non-zero Basel rate of production. I set this B constant equal to 0.5. If I set the Basel rate of production to 0, then the low value will go to 0. All right. And if I change B, I can ask two questions. Do the fixed points change? Does the high value of A and the low value of A change for the different initial values of B? And clearly the answer should be no. Although the time to convergence also should stay the same. And so as I move that initial value B, I select whether I go high or low, but it doesn't change the final outcome. All right. Now, what about changing the Basel transcription rate? If I change this little lowercase b, does that change the fixed points? Should it change the fixed points? Should it change the value of A star high and A star low?
I think it will change.
Okay. So that's the first one where it might be worth trying it out. So the answer is yes, it does. It changes both the temporal behavior and the value of the fixed point. In this case, if I have no Basel transcription, for this particular set of equations, I only have one stable fixed point, which is everything is turned off. And you'll see this set of patterns. So why don't we actually take a minute, because you'll need this. If you have the existing code, if you can, take a minute and see if you can redo it with this set of equations. So it'll take maybe 5 minutes to retype them. There should be a copy, or there should be a copy of this. Let me see if I have this actually as a, it should be available in the student materials folder to produce this code. If I don't think I have it as a, let me check to see if I have this also as a Colab notebook. I should probably make a Colab notebook for it. Sorry, I copied that one into the student materials folder for you. I realized I didn't have it copied there if you want it. But it doesn't take that long to retype it yourself.
Yeah, I almost finished.
I haven't talked about how to do function definitions in Antimony, but this is a case where a function definition might be useful. If you define a hill function h of a comma k comma a comma h lowercase h, hill of a comma k comma lowercase h, then you can type these equations much more rapidly. But this lecture 8 toggle initial value scan I put in the demos folder for lecture 9. So if you want to run this.
What's the initial value of K? Oh, sorry, I didn't see that. 0.5, 0.7. A2 is 1.
Now, what's a little bit interesting here when you get it to run is that in this case, both A and B go to low values. You don't have enough basal production to make anything work. But if you look at these B equals 0.18, you'll see that the value of A will drop rapidly and then hang out around 0.5, 0.6 for a while and then go to the low value. And that is happening because you're getting near a bifurcation where you get a second stable fixed point. And so this is what you call a ghost of a fixed point. It's not there yet, but you're getting close to it. And I don't want to spend too much time on this one. I do want to have the code, the basic equations there because I want to use them for some other things in a minute.
Same result.
I'm sorry, did you have a question?
No, I have no question.
Okay, good.
It's running.
So you got it to work?
Yeah, it's working.
Okay. And I was just looking, I do have a collab version of this. Now, if we put b to be big, so change the value of lowercase b to be 1.2 and run it again, and now you'll see you should get a high value for both a and b, regardless of the initial condition. Let me know if that worked for you. So all you're doing is changing the value of lowercase b to be 1.3, 1.5, so on.
Yeah, I got it.
Okay, great. Okay. So it winds up that if b is intermediate between 1.185 and 1.2, then you actually find three values of a star, a high value, the low value, and then the unstable fixed point when a and b, initial value of a and b are equal to each other. So the next exercise is, so here are the situations for a at least. If b is small, a goes to a stable low value. If b is big, a goes to stable high value. If b is intermediate, then if a is bigger than b initial, I go to a high value for a. If a is less than b initial, I go to low value, and if they're equal, I go to the unstable steady state. So the next exercise would be plotting the bifurcation diagram. And you may have already done that. If you haven't, I have the code for it here. But I'd like you to sweep the value b and then plot the steady state values of a and b as a function of b, of little b, for b between 0 and 2. Okay, but here you want your x-axis to be the value of B, and the y-axis to be the final value of A.
Oh, that will use the
So
function.
this is a bifurcation plot.
Oh.
So I think we did it, what, 2 weeks ago? We
Yeah,
did this for the oscillator one. So
but the
so that
function,
you should,
the function that was used to find the stable state was not stable. The function itself was not stable.
right. So in that case, just run the simulation for some long amount of time. Run it for a few hundred time steps
Yeah.
until it's equilibrated. That's why I always suggest running it a few sample simulations to get a sense of how long things take and what the values of the variables are, so that you know how to set the time scale and the length and the amplitude scale. For this one, get steady state values should work, but I can't promise. I'm doing it myself here. So checking it myself. Well, so for me, get steady state values fails if, well, actually for these parameter sets, it's okay. For the ones I was looking at, it failed. If the coupling strength I is too small, it won't work. But for B scan, it should be all right. What did you get? Okay, so in this case, that's getting there. So a couple of things, I'm a little puzzled by a few things about this, but let's look at it together. The first thing is you probably need to make your step size for B smaller. And I realize the simulation takes a while. And you're probably going to have to make B go to a value bigger than 2 because you haven't seen the upper bifurcation yet. Let me show you what I got. This is what I got.
Oh.
It's possible some of your parameter values are slightly different from the ones that I have. Okay, so yeah, there we go. That looks better. How long are you running to get to the fixed point?
300 points seconds, I think.
So, and you're scanning B. So I'm actually puzzled by the shape of what you're getting. It looks to me like it's not converging. So are the parameter values you're using the ones that are here? H equals 4, K 1.5. K 1.5 doesn't quite look right. K equals 0.7, K2 equals 1, I equals 0.6.
Yeah, they are the same.
Let's look at your code.
Oh, what's the initial value of A and B?
Well, if you want high, then it shouldn't matter as long as A is above B. Get the high one, and the low one you want A to be below initial value B. But let's look at your code together because I'm pretty confident that this curve is right. So if you're getting something different, I'd like. You're definitely seeing the bifurcation, but I'm trying to see why it's not. Okay, let's see. A to the H. Oh, I see it. Look in line 591.
591.
Look in the denominator of the second term of that term. Should that be A or B?
Wait, what? It should be oh, the typo.
Yeah.
Okay.
Right now.
Okay, let's try again.
There. Okay. So now you can limit the max value B to be 2. So sweep the value B between 0 and 2.
Okay. Here.
I mean, it's certainly not that I'm never wrong, but I've been I've been working with these long enough that usually I catch these things. I do make mistakes. And then the other thing I would do, it'll run a little slower, but I would make the step size for B smaller because something interesting happens. For the low value of B, it's a subcritical bifurcation. You see it jumps. You go from the center value. It doesn't, on the high side, it comes back smoothly when it turns off, but when it turns on, it goes boom. And so there really is a jump. Okay. And you're not quite converged, so you want to run to 500 time steps or even 1,000 time steps. Also the 300, make the 300 500 there too. the third one is the sub step, which is okay. Yeah, there you go. There you go. Try that. Yeah, there we go. That's better. You see that little sort of mustache shaped thing where the blue and the red line, the orange line come together on the right? Yeah, there. That's because you're not quite converged.
Hmm.
If you ran it for a very long time, that would disappear.
What about?
Yeah, that should get rid of it.
Yeah.
So, okay, great. All right, so let us come back to my screen share for a second. So now that what we had was we had the stable fixed points. So how do you think you can get the unstable fixed points? They're actually, in this case, there's a cheat to get the unstable fixed point because you know what it is. The other way to get the unstable fixed point is to multiply the rate loss by -1, which runs time backwards. Did you try doing that to find the unstable fixed points?
I haven't tried.
Okay, so why don't you try that? So basically in your rate laws, put a -1 in front of everything. Multiply the whole thing by -1, and then run it and see if you get the unstable fixed point. The trick there is you have to pick your initial conditions pretty carefully because if you go too far out of the range, it'll blow up. You have to be within the basin of attraction for the unstable fixed point. So that'll only work in this middle region for the high and the low values. There is no unstable fixed point, so it'll blow up. There's also a simpler way to do it, which maybe is easier in this case. How can I find the unstable fixed point? What's the condition for the relationship between A and B for the unstable fixed point?
I got a upside down figure.
Let's see. You want to share it or let's see.
Yeah, it's in the chat.
So that is not what I would expect. So let's see what you change. What did you change in your code? So you changed you changed the time direction and you got that. All right. Well, let's not fuss with that right at the moment. We can come back to it. What's another way to get the unstable fixed point? What's a condition for the relationship between A and B to be on the unstable fixed point? We go to the upper branch here, the top branch. Am my screen sharing?
I cannot see the slide.
Sorry about that. Okay. So we go to this upper branch here when A is bigger than B. We go to the lower branch here when A is less than B. So what happens if A equals B? That should give us the unstable branch. So why don't you, instead of trying to do time reversal, redo this plot and add a plot where you start A and B equal to each other and plot that and see as a third line and see what happens. Okay? Does that make sense as an exercise?
I'm not sure what to do here.
So
So look at your code.
yeah,
Your initial code is starting with A bigger than B, and that's your orange line, and A less than B, which is your blue line. What if you plot a third line where A and B initially are equal to each other? Start with A equal to 0.5, B equal to 0.5, and run it and see what happens.
okay, yeah, I will try.
Now, if there are numerical instabilities, what you'll find is that it's going to pick either the big one, top one, or the bottom one. But if the integrators are working well, you should stay on the unstable fixed point. The time reversal will always work, but the cheat.
Yeah, it shows the unstable region, but I'm not sure it's the figure you want.
Okay, so here what you're seeing is okay. So you are seeing the unstable one. So what you're seeing is that sometimes it's drifting to the top or the bottom.
Yeah.
Which is what I was saying about the numerical stability. How long are you integrating for?
The time span,
Yeah.
1,000.
Yeah, so you might make it shorter and see if it works better for that. Because in fact, analytically, if you start with A equals B, you should always stay on that unstable branch. But what you're seeing here when you're jumping to the top and the bottom is that the integrator has got noise in it, so A isn't equal to B anymore. So that's actually telling you that the integrator isn't perfect. So it's a little odd actually if it's symmetrical. And let me look at my old code and see how I did it. I don't remember how no, you're still getting noise. If you're getting noise, then
Okay, if I simulate it from 0 to 10, it's okay.
okay, so that's probably good enough. They won't be perfectly converged, but it's probably close. I'm not sure what happened with your with the one where you did the time reversal. The time reversal will work. So why don't you now that you're using the initial values equal to each other, now try doing the time reversal and see if it should give you this that unstable branch.
Okay.
It's easy to accidentally not multiply everything by -1, in which case it won't work. You have to really multiply everything by -1.
Yeah, all the dv dt should multiply it by -1, right?
Everything on the right-hand side gets multiplied. So the decay term turns into a source term. Instead of minus K2A, you'll get plus K2A. And then instead of plus K times everything here, it'll be minus K. If you leave the decay term turned on as negative and the source term is negative, then it'll get different results.
But the solver says it cannot converge.
Right. So this is an issue. If you start, so if B is less than this initial branch, there is no unstable one. And that means when you run it backwards in time, unless you start with A equal B, and you run it backwards in time, you're going to get something that diverges. And so what you have to do in that case is you run it for, say, 10 Monte Carlo time steps. And you say, if the value of A gets to be big, or if the value of B gets to be big, then you stop. And you say there is no unstable 6th point for that. So it takes a little bit more work to get that to work for the -1, because the system is intrinsically unstable. The stable fixed points have become unstable. The system before, when you have first order decay kinetics, the system is automatically stable. It never diverges. The values don't diverge. When you run it backwards in time, the values can go to infinity. So you have to catch that. You have to trap them. Maybe we'll leave that as a homework problem exercise for you to develop that a little bit.
Okay.
But it's a useful thing to do.
So I have to write some logic in the Python script to determine whether it's stable or not.
That's right.
Okay.
So you run that, you flip the value of A and you flip your sign here. Okay. For your initial conditions, you know that the unstable branch has A equal to B. So you start with A and B equal to each other. Your goal is to try to get rid of these the noise that you're seeing in the simulation that you showed, where the solver noise is throwing you off. That's why you want to run backwards. So try the one running backwards in time where you start with an initial value of A equal to the initial value of B. Does it still diverge on you? I guess it will. It will if it will for B less than this B less than the 0.27 or bigger than 1.1. Could still do that. Try it though.
I'm getting weird results.
What are you getting? Put it in the chat. See.
This is 10 time steps and simulated from 0 to 10.
Well, A, you notice it's negative.
What
Right.
is negative?
Look at the y-axis.
Yeah.
Well, your values are supposed to be between 0 and 1, and here they're between 5,000 and -12,000.
Yeah, but the initial values are still both A and B starts from 0.5.
So my guess is that you haven't multiplied your rate laws by -1. It's somewhere something isn't flipped. I'm happy to look at the code with you if you want.
Yeah.
Okay.
Everything is multiplied by -1.
Okay, you multiplied the decay by -1. Looks like it's okay. You multiplied everything by -1. That looks good. Okay.
Yeah, and I didn't change anything except for this time step here.
So let's start out by just going back up to the top. And let's not, instead of running the loop, let's just run initial value A and B are equal to 0.5. That's fine. Let's just add line 603. Let's just simulate it for a time 10.
Okay.
And then just do a R dot plot and see what we get. So we'll plot the time series. Yeah, it's good. Okay. And let's see what we get with that. So you see it does, so it blows up, right? It blows up after, it blows up pretty fast, actually. So that means our initial values of A and B are a problem. Okay, what's our little b? What was our initial? What's little b was 0 here.
Yeah.
Okay, so let's make it 1. Run that. Still blowing up. Still blowing up. Let's see. Why is that blowing up? That should work because we are within the basin of attraction of, for 1, our initial value of A and B should be about 0.6. Let's start our initial A and B to be, let's see, let's make sure everything is the same here. 0.51. Huh. Okay, multiplied everything by -1. Oh, I know what it is. Look in line 591. Hold on a sec, please. Maybe it's, no, it's okay. My mistake. My mistake. I thought I saw a typo, but there is no typo. You were right. Huh. Let's try starting with A and B equal to 0.6 instead of 0.5. Still blowing up. Interesting. And little b is 1. Huh. Okay. I'm afraid I'm not gonna be able to solve that problem now. I genuinely don't know why that's not working for you.
Yeah, I don't know either. That's
So I apologize, but I don't have. Let's see if my code, does my code do the unstable branch? No. All right. Okay, I think I'd rather, I apologize, but I'm gonna let you, you're good at this sort of thing. I'll let you play. You can look at some of the code that I've got online and see if you can get that to work. Because that should be, the time reversal one does work, but it does require some fiddling. I'm not sure why it's failing for you. Let's do one last thing before we just start with A equal 0.6 and B equals 0
okay.
and see if it still diverges in time 10 time steps.
B starts from 0.
And see if it still blows up, then we'll give up.
It still blows up.
Yeah, the time reversal thing is a little bit, as I say, is a little bit fussy, and because you have to pick an initial value that's quite close to the unstable fixed point for it to work. But again, since we know that the initial values of A and B should be equal, and we know actually from looking at the picture what they should be, it should be possible to figure it out. The way you normally do it is the following. You run this forward, and if you find 2 stable fixed points, this A high and the A low, then if you pick the initial values of A and B between A high and A low, then when you run it backwards in time, you should get the unstable branch. If you don't have a separated A high and A low, then you can't do anything. Then you're stuck. And that should work. And I'm puzzled by why it's diverging for you. I'd have to play with it. Okay. So let's come back to the phase plots. You had code that did phase plots, right? You run the phase plots before.
Yes.
Okay. So why don't we do a phase plot of this circuit? And to begin with, just set I equal to 1, the maximal inhibition. And there are going to be two kinds of phase plots. We did the flow diagram, right? Where we start with initial values and we plot the time series. Did we also do one where we did little vectors at each point? Yes or no? I don't remember.
We did that. We did that.
We did both.
We did both, yes.
Okay, so let's take a look at this. So let's look at this, get rid of your minus ones. And let's look at the flow for this circuit together for I equals 1.
Okay.
And also if you can do the one with the small vectors, that's also interesting. If you don't have that code, I can give it to you. I have it pulled up. And you'll notice from this picture, I did not use Tellurium. These are old slides, and this uses XPP odd. And so we need to figure out a way to plot those null lines. XPP odd plots the null lines for us. And I don't have the null lines automatically plotted. And I know Connor wrote code to do it, and I can't. I keep meaning to look for it, and I haven't found it yet. Okay, let's see. All right, so here's a case where thinking what you've got here. Let's look at this together. What you've got here is not a bad start, but you'll notice that the fixed points are between 0 and maybe 2.
Yeah, it's too
So
large.
So it's too large. So why don't we change the range to be between 0 and 3, for example, to begin with?
Okay.
Maybe it'll zoom in even a little more than that. Okay, that's better. We might even try to zoom in a little more. Okay, so why is the fixed point that you're getting about 1.5? See, this is what I'm puzzled by. The values for the equation, for the parameter sets that I've given you, yeah, I guess the fixed point's a little bit 1.4, so that's not too bad. So that should be all right. So this is pretty much what we expect. So you have two stable fixed points, A equals 0 and B equals 1.4, and A equals 1.4 and B equals 0, and an unstable fixed point at 0.5 comma 0.5.
Mhm.
Now here you're plotting the flow. If you plot the vectors, you get a slightly different result. That's for I equals 1. Now why don't you try redoing that for I equals 0, I equals 0.2, 0.5, and so on, and see what happens. And I've already plotted the bifurcation diagram here, so you actually know that the bifurcation happens when I is about 0.3.
Okay.
So basically, if you make the value capital I less than something like 0.1, you'll have a different result. If you make it 0.8, it should look like what you got for 1. And around 0.3, things are interesting. And actually, looking at the figure that's on the screen, I realize that my time reversal argument isn't going to work in this case. Because the unstable fixed point is a saddle. And if you time reverse a saddle, it's still a saddle. So that unstable fixed point stays unstable when you time reverse it. So my instructions to try time reversing it won't work. But you should still be able to get a stable fixed point at 0, which you didn't see. So I'm puzzled by that. Actually, maybe not. Let me see. You go to no, 0 isn't a fixed point. So no, I'm wrong. Because B is non-zero. So if you time reverse this, if you started out with A and B bigger than a half, you'll get infinity. And if you start less than a half, you'll get minus infinity. So your result was right. So
Okay.
the time reversal method won't work to find the unstable branch here.
But why?
Well, we could actually do that. Let's do it. Let's actually do it. Take the code that you have that generated your flow diagram here. Put in the -1s and run it again and see what happens. And plot exactly the same thing. See what happens. It might be better to plot the what I have here on the screen, the vectors, rather than doing the flows the way you did it. But you can try either way.
Yeah, I can see so the flow is pointing outward.
Right. Oh, so before you go to there, the one you're showing me here, which you shared here, was different values of I. So let's
Yes,
see
yes.
the normal.
I tried to
So for I equals 0,
use.
everything converges just to they're independent, so they both reach their stable equilibrium. Okay. When I is 0.4, you get I 0.4 still looks like it's giving you a single stable fixed point. I would have thought that you would see the bifurcation. So before you flip the sign, well, let's see. So let me just show you what I would get when I change the values of I. This is the forward one before we time reverse it. These are what I get. So if I is 0.38,
I'm sorry, I cannot see the slides.
I'm sorry, we're still back. Sorry, my mistake. There we go. So you got the screen share okay now?
Nope.
Huh. Now?
Still nothing.
Huh. All right, let me try that again. It's telling me I'm doing the share.
Oh, now it's okay.
Okay. I don't know what happened. It showed me the screen with the share. So, but obviously when I canceled the old share, it didn't actually cancel. Oh well. Okay, so here, when I'm below the bifurcation, 0.3, 0.2, I have a single stable fixed point here where A equals B, and the value here is about 0.7. At 0.5, I'm on the right side of this. I should have an unstable fixed point in the middle, which I do. And I have a stable fixed point on A equals 0 and 1.4, B equals 1.4, and B equals 0, A equals 1.4. So you get those two stable fixed points. If I look at the null lines, at 0.55, they're almost tangent to each other. As I increase the value of I, the region in which the volume in which they've crossed in this little bow tie gets bigger and bigger. And you'll notice also that the values of the stable fixed points move apart. Here at 0.57, the stable fixed points are not at 0. I said 0 and I was wrong. They're actually here and here. And as I increase the value of I, they move to 0. For I equals 1, they go to 0. So I see them diverge like that. Now, if I flip the direction of these, if I flip time, these vectors will flip direction. They go the opposite direction because these are the flow in time. And so what happens here? This, for small values of I, this stable fixed point becomes unstable. And so my flow is outward everywhere. And you'll notice that if I go to large values, I'm going to flow out. If I go to small values, I'm also going to flow out. So there's nothing that's going to confine me to not crossing 0. So I'm only going to have negative values. So I'm going to be allowed to have negative values. And so I'll have no stable fixed point in this case, because the null lines aren't going to change. The null lines are invariant when I flip the direction of time. If I flip the directions here in the, say, I equals 1 case, if I'm in the upper right-hand quadrant, I'm now moving away and my values will diverge. If I'm in my lower left-hand quadrant, I'm moving down and to the left, so my values will diverge. The only places that I will be let's see, am I even trapped there? If I start between my null lines, I will be moving down. If I move between the green and the red null lines, I'll be moving down and to the right. And my unstable fixed point should become stable. So I have, but it's a very limited range of values for which that's the case. So why don't you see, what happens when you reverse the time and you do this plot? What do you get for I equals 1? Let's see what we get.
Equals 1. Okay.
Okay. Let's see.
The flow is
That was actually what I was expecting, which is why I was confused, maybe confusing myself a little bit, which is, let me put this screen share it so we can talk about it together. Let's see, make sure I'm screen sharing the right one. B. Okay, my screen sharing your plot.
Yeah, I can see that.
So my initial stable fixed points are now unstable. which is what I expect. My initial unstable fixed point is still unstable when I've time reversed because it was a saddle. And so the direction that was stable is now unstable. Direction that was unstable is now stable, but I've still got one stable and one unstable direction. And so, yes, so if I start with A and B large, everything goes to infinity. If I start with A and B below this curve that connects the former stable fixed points, things go to minus infinity. So the time reverse system has no stable fixed points. So I was correct when I said that. I was incorrect when I said that time reversing would work. I was correct when I corrected myself and said time reversing won't work because it's a saddle. So I got that. I outsmarted myself on the time reverse. So what you've got here actually is what I would expect. So to find that unstable fixed point, it requires a more effort than just doing the time reverse. In one dimension, the time reversal always works, but in 2 dimensions it doesn't, because you have, as I say, this is because of the fact that in 2 and higher dimensions you have more ways to be unstable than stable. In 1D, if you start with an unstable fixed point and you time reverse it, it is by definition stable. In 2D, if you start with something with 2 positive eigenvalues and time reverse, you get 2 negative eigenvalues and then it's stable. But the saddle stays unstable when you time reverse. So that was my mistake. All right. So, and these were the flows that I got, which are pretty close to the ones you showed in your picture. All right. Well, I think we're out of time for today. I wanted to do the stochastic case, and why don't we'll do that next week. That's where we'll start next week.
Okay.
But something interesting happens, which in the stochastic case, and you saw actually something a little bit like that when you tried to find the unstable fixed point. And sometimes you got the unstable fixed point, and sometimes it jumped to one or the other. What you're going to find when you go to the stochastic simulation is that you jump back and forth between the 2 fixed points. The system will sit on one, and then it will jump randomly to the other one. It'll sit on that for a while and jump randomly. That's called chatter. And that is biologically important, because if you want to have differentiation and you want the differentiation state of a cell to be stable, you can't have chatter. You don't want the cell to spontaneously switch its type at random times. And what that is going to tell you is actually that this kind of gene regulatory network cannot be responsible for stable differentiation of cells. Because if it did, your cells would de-differentiate or trans-differentiate all the time. You wouldn't be able to build multi-cell organisms. Okay, anything else you want to go over now before we actually started a little bit early, so it's actually a little overtime today.
I have no question.
Okay, thank you for being patient, and see if you can come up with an algorithm. You can look at my old code, but see if you can come up with an algorithm that'll track that unstable fixed point. The other one is, and I don't have the code. See if you can do a little bit of digging and see if you can find an algorithm that will give you those null lines. In principle, the null lines are pretty simple. You write the right-hand side of this equation, and then you ask the question, for what values of A is the right-hand side, A and B is the right-hand side 0? In practice, it's not so simple to do that evaluation. But there is code in SciPy that'll do it. And so if you do a little bit of reading, you should be able to figure out how to do it, to plot the null lines. Okay?
Okay.
Okay, great. Thanks so much.
Thank you.
I'll see you we said Thursday, right?
Yeah, Thursday.
Great. Okay, look forward